Let's see now how we can interpret the data. An experimental design is made of data and metadata. Metadata is all the data about the data. So you can put in this category a lot of things, like when you did the experiment, who did it, and all the information about the biological material, what genotypes, where did you buy them, how many, when did you sow the seeds, and when did you collect, and so on and so on and so on. So the more information you have, the more metadata, the more rich will be then the interpretation of your results. Within the metadata, there is a particularly important thing, which are the factors under study, what we call the controlled variables. For example, the genotypes, the treatments you made, and then it gives you some modalities. Here, if you have 3 factors under study, then you will have, and you have 2 modalities, like for the variable 1, you have wild type and knockout, a mutant. Variable 2 treatment, you have control and hormone treated. And the third treatment is low light or high light. And if you have 5 biological replicates of each modality, then you have 40 samples. And on those samples, you make some measurements, like metabolomics, and you have a data set. And this is just only by crossing data and metadata that you can obtain results and interpretation of your experiment. So metabolomics metadata are what, who, when, how. Genotypes, location of the experimentations, material protocols, available personnel, crop intervention, temperatures in the phytotron in the field, names of operator, climate, type of equipment, the different anomalies you found. Maybe there have been an air conditioning failure at an early stage of the growth of the culture of the crop, etc., etc. So this is typically what you have in the laboratory notebook. Now let's look at a very simple data set to understand this data formalism. You have here 2 variables, age and weight, measured in 20 individuals. And these individuals, you can label them according to their sex. So it's a bimodal variable. They are either men or women. And so you have measured data, measured variables, and controlled variable, the sex. The controlled variables are named Y, here in green. And the measured variables are noted X in blue. The table is organized as follows. You have individuals in rows, maybe dozens or sometimes, and quite often, hundreds of individuals, which means samples. And then the variables in column, starting with the controlled variables, so the metadata that will be necessary to interpret the results. And then finally the metabolites, a dozen, hundreds, or sometimes thousands, not of metabolites, but of ions, before you can regroup them into metabolites. So basically the Y matrix and the X matrix. So you have n individual samples, k measured variables, so named x, x1, x2, x3, etc., and m controlled variables in the matrix Y. They are also called the descriptive variables. The measured variables are continuous, these are signals or concentrations, while controlled variables are usually discrete, these are the classes of samples. The treatments applied. So it's a description of the samples. So the signals in the variables, well, usually it is in mass spectrometry, the extracted ion chromatogram peak area. So it is a pair of retention time and also mass to charge ratio. In NMR, you will have chemical shifts and peak areas. So most of the time anyway, it's peak areas because the idea is to quantify these compounds. More rarely, and rather in targeted analysis, the metabolite concentration, absolute or relative concentrations, in micromole per milligram of sample or in microgram per gram of sample. The individuals are sample extracts, so you have, for instance, 108 samples because you have 2 organs, 3 genotypes, 3 treatments, 3 biological replicates, and 2 technical replicates, for instance. So you can decompose the samples according to the classes of treatment. Now, to understand this data, we use multivariate statistics. The principle is as follows. So univariate statistics, usually when you have less than 6 variables, you can do a manual analysis of these variables. And in this case, you have more individuals than variables. You can use univariate statistics like parametric tests or non-parametric tests, like ANOVA, analysis of variance, student test, regression analysis. The good aspect of this is the threshold, the risk you take. We call that type 1 risk, to declare that there is a difference that in fact is due to random distribution. This is the famous p-value. So this is type 1 risk. It's a risk of false positive, and you can set a threshold like 1% or 5%. And if the threshold, if the calculated p-value is inferior to this threshold, then you consider that you take for granted this result. In multivariate statistics, usually you have more variables than individuals. And you can use different tools like the principal component analysis, PCA, but also partial least square analysis like PLS, PLSDA, OPLSDA, which is orthogonal partial least square discriminant analysis. And finally, HCA, hierarchical clustering analysis. We will just focus on principal component analysis and orthogonal partial least square discriminant analysis, PCA and OPLSDA. The PCA will give you the true pattern in your dataset. It is very interesting to find the trends of your dataset, to understand, to feel the quality of the information within your dataset, and especially to detect outliers, which are samples that are very different from all the other samples, especially they are very different from all the other samples which are from the same conditions. So in this case, it can mean that this sample is an error. It has been treated badly. Maybe there is something happened during extraction or analysis. And so it's a great tool also to sort the samples, to remove all the very non-typical samples that are introducing noise in the dataset. On the other hand, the OPLSDA will make a classification of the samples according to a criterion you decide. For example, you may want to see the differences between control and treated samples. So it's very useful to quickly extract the potential biomarkers. And that is one great advantage. You can do that with the PCA, but it's much more straightforward with PLS in general, and it's very efficient with OPLS. So multivariate statistics are performed by algebraic calculation that result in the construction of what we call latent variables, the components. And these variables are synthetic variables. They are combinations of the initial variables, the metabolites, in a way that they will summarize the information from the dataset. And the result is then plotted in 2 dimensions or 3 dimensions, it depends, on the principal component or the calculated component rather than a projection of the variables themselves. The advantage is that you can study simultaneously many variables or even all of them. You don't lose any information at first, and it allows you to study correlation between variables and to highlight similarities and dissimilarities between individuals. Let's have a look first at the principal component analysis, PCA. So the calculation of the main components, the principal component, also named PCs, is made as follows it's algebraic calculus. I won't get into detail for this also because it would take too much time. But these principal components are linear combinations of the initial variables. And they express a decreasing and independent part of the variance. We say that the principal components are orthogonal to each other. So, for example, the first principal component expresses a maximum of the variance of the dataset. This is also what we call the maximum inertia of the cloud. And the second principal component expresses an independent part of the variance lower than the first, and so on. And here you have a plot showing, well, it's a histogram that shows the percentage of the total dataset variance that is expressed by each principal component. So the first principal component here expresses more than 40% of the whole variable. The second, around 20, the third, around 12, and so on. And if you make the sum of all these percentage explained, you have 100%. It means that on the whole set of principal components is shown the whole variance in the dataset. But what you want is to look at the main sources of variation. This is what you want to detect with the principal component analysis. So you will focus on the first 2 or 3 principal components usually. So as a result, there is a large table of data that is transformed into 2 smaller tables. What we call the coordinates of the individuals on the principal components. It's called the scores. Basically, it's just the projection of the individuals on the variables that has been calculated, the principal components. And also, you can calculate the coordinates of the initial variables on the principal components. We call that the loadings. It will indicate how much one given initial variables participate to the calculation of a principal component. And finally, we can project the information on a score plot and a loading plot. So we go from a table to a graph. The score plot shows the individuals and the loading plot shows the variables. Interestingly, principal component analysis is an unsupervised analysis. The calculations do not incorporate any working hypotheses, such as expected differences or similarities between samples. You don't say anything. It will show you the objective structure of the data, and this is very, very powerful. The matrix, another way to say that is that the matrix Y is not included or used for the calculation of the principal component. You will use the Y matrix, that is the descriptive variables, just to project, to label the individuals on the plan. And you will see that some samples cluster together when you do that, and because they belong to the same class of samples. They have the same value for any given control variable. So principal component analysis is a very interesting starting point for almost all metabolomics analysis. It provides you with an objective view of your data. You can detect outliers, atypical individuals. You can evaluate the information potential of the data set. If you identify well-separated group, it was the case in the previous slide, of individuals, it means you have a lot of information to grab, to catch from this data set. You can also very quickly identify the variables that have a positive or negative effect on a given class of samples. To make it clear, I will show an example. Very simple. So this is an investigation of the breakfast eating habits in Europe. So you have different countries in the first column and different foods in the following columns. And then you have the frequency of consumption of these different foods in these different countries: Germany, Italy, France, etc. When you make a PCA on this table and you project the score plot, which is the left graph, you see that the individuals here are the countries, and they are projected, they are clustering on the graph. The abscissa is the first principal component. and it tells you that it is expressing 37% of the total data variance. The second is the principal component number 2, and it expresses an independent 21% of the variance of the dataset. So altogether, you have 37 plus 21, which is 58% of the data variance expressed in these two principal components. So you have the major part of the variance, which is the useful information of your dataset, expressed in this plan. And on the right graph, you can project the loading plot. And this time, on the same map, you project the initial variables, which are the foods. So the coffee, potato, butter, and so on. So this is what is expressed here concerning the space PC1, PC2, expressing 58% of the total variance. And what is very important is that you can interpret these graphs geographically. You don't need any other information, or you don't need any quantitative data more than that. If you have samples that cluster together, it means that they are almost the same. They are alike. And Sweden and Denmark here are very close on the map, which means that their habits of breakfast are nearly the same, and they are northern countries. So it's not very surprising. They have the same, they are culture with a lot of family bond, in fact, as a culture. And of course, you will find also Finland and Norway, and then you will find France at the bottom, and Italy and Spain. So here, in fact, on this map, you separate Germanic culture in Europe and Latin culture. And you can associate exactly in the same way the food to these different cultures. And for example, fish and vegetable and crisps are more often consumed in the northern country like Sweden and Denmark, while garlic and olive oil is more specific of Italy and Spain, which is no surprise, of course. So it's very easy and very fast, a very fast process to understand this data. Now you can explore a bit more of this data set by plotting then on the third principal component. So if you look at the ordinate here, it's PC3, the third component, which expresses only 9% of the total variance. But it concerns mostly Ireland and England, which is quite interesting because they belong to the same type of culture. They are very connected. And you see that they are associated with less consumption of grain coffee, but a higher consumption of tea and jam, which is, of course, something that is famously known from the English breakfast. So I hope this clarifies you how to use a principal component analysis, even if we don't see how to calculate this. It's a bit complex, and it's not the point here. The point is to understand what you can do with the principal component analysis. Another very useful thing is to do outlier detections. Here you see you make your principal component analysis from your table. You don't know anything about your data set at this time. And you see that 1, 2, 3, 4 individuals are very, very different from the rest of individuals. They are outliers. And outliers, you can rationalize the outlier property by using a confidence interval defined by a multivariate generalization of the student test named Hoteling's t-square. So these metabolites, these samples are weird, they are strange, they are very different from the others. So now you have to investigate why. And you can maybe try to find the variables associated to this discrimination. And maybe something went wrong for one of these measurements, or maybe this is a result, an interesting result, because you have atypical samples from atypical situations. So they usually point out to problems during the experiment, the sample collection, the extraction or analysis, or the data processing. So either you investigate and you fix the problem, you correct the data because something went wrong. Maybe sometimes you have to reanalyze the same compound. So it's usually safer to use a backup replicate. So to have something in the fridge or in the freezer, you can a backup sample. So it's exactly the same sample, but it's a small amount you put aside to be able to use it later. And in this case, it would be very useful to see if it comes from the sample or just from an analytical problem. Or, so this is the hard way, but the easier way is to just eliminate the sample from the analysis and do the principal component analysis again. And this time you find that there is no outliers. So maybe this sample, something was wrong with it, and you can reasonably discard it from the analysis because it is atypical and it's not going to help you understand the biological processes in your experiment. What matters, in fact, is to find the clusters. So for this, you will label your samples according to the Y matrix, the control variables. So in this case, for example, you have different type of mice. You have white, black, nude mice. So nude mice are orange, white are gray, and black are black. And you see that in this experiment, the metabolites were very different in these different genotypes of mice. And you can find markers that are associated, for example, with black mice. And these are pairs of retention time and mass to charge ratio. So this particular biomarker is a biomarker of black mice. And if you go to the bottom of the loading plot, you will find other markers that are associated with nude mice. And on the left, you'll find biomarkers associated with white mice genotypes. Let's talk now about OP-LSDA. So in that case, difference with PCA is that OP-LSDA is a supervised analysis. And it's more a model construction than just an objective description of your data. The idea is to build a model that will maximize the separation between two groups you defined a priori. Example, well, white mice and black mice, or nude mice and white mice, and so on, or control versus treated. So this time, you build the model in two steps. First, you calculate the model on a subset of the individuals, and then the model is tested on other individuals that did not contribute to build the model to determine if it's efficient at classifying the samples. So we say that you have a training set of individuals and a testing set of individuals. So in this case, OP-LS is supervised. You will use the Y matrix to calculate the components. It's a kind of regression and prediction method that extracts information from the data X that is related to information known in Y. It's a modelization. So we could say that the PCA will show you the objective structure of your data, while the OP-LS will force the model to build axes that really separate the two conditions you want to separate. In this case, the blue dots and the white dots. So this is typically the compared results you can have with a PCA and an OP-LSDA. With the PCA, well, the samples are somehow discriminated because, well, the data were like this. So it's an efficient experiment with a lot of things to investigate because you have a clear difference in the metabolome of the black individuals and the red individuals. But with the OP-LSDA, it's even more obvious. The first component is really helping you to discriminate the variables that are responsible for this discrimination. The discrimination is much stronger. To evaluate the quality of an OpenSDA, you have two types of figures. You have first the R square, which is the goodness of fit, also named R square Y. And you have the Q square, which is the goodness of prediction. R square is the variation in Y explained by the model. So it is its recognition capability, not to confuse with R square X, which is the percentage of the total variation of X that is related to Y. So R square Y, if it is 97%, it means that 97% of the difference between the groups that you designed, that you wanted to separate, is explained by the model. But the R square X is only 24%. It means that 24% of the variance of the dataset is related to the difference between the two groups studied. So you see, different notion. One concerns only the discrimination you want to look for. The other is this discrimination as compared to the global variance contained in the dataset. And finally, the prediction capability, Q square, which is also a cross-validation test. I said the model is tested on data that were not used to calculate it. So you can look at the efficiency of classification, the amount of sample that is correctly classified. And so if you have Q square is 91%, it's a very good model quality. So the higher R square and Q square, the better the model. So you have these score plots where you discriminate OpenSDA in the OpenSDA, but you also have the S plot that you can use. This is a projection where in abscissa you have the magnitude, which is the covariance with the model classes. And then you have the reliability in ordinates. It is a correlation with the calculated components, positively or negatively. It doesn't matter. So you have a typical S plot like this, where in the middle you have noise. So these are variables that are not discriminating the samples under investigation. On the top right, you have biomarkers, and on the top left, you have on the bottom left, you have also biomarkers. So the only difference is that they will have values, opposed values in the different samples. So the biomarker on the top right will have strong values maybe in the condition A and low value in the condition B, while the biomarkers in the bottom left will have high values in the sample B and low value in the samples A. So this is an indication of the discriminating variables. So well, you can define high-risk zone. Well, these are the zones of this graph where you are not going to find interesting variables. They are very likely to be not really discriminating. And then you have low-risk zones in which, well, you can find the few very discriminating variables. And then you have a gray area where you have some risk and maybe you need to check with other means like univariate statistics if indeed these variables are really discriminating. To do all these multivariate statistics, there are many, many tools. I personally love Metabolomics because it's an online software, it's free, and you can have many, many very useful statistics that are quite simple to use and very straightforward. It's a very straightforward way to explore your data. A major step in data analysis is the molecular structure elucidation of the biomarkers. First, let's talk about identification criteria. To identify a molecule, you need to know its structural formula and therefore implicitly its raw formula, its molar and monoisotopic mass, and its common name or according to the chemical nomenclature. So for example, here we have chlorogenic acid, which is also named 3,0-caffeoylquinic acid. Its molecular formula is C16H18O9. Its molecular weight is 354.31 gram per mole, and its monoisotopic mass is a bit less. And well, we have its structural formula, which is the organization of the atoms and their bonds in the molecule. So first, let's examine the interest and limitations of measuring the exact mass. We've seen what it means. And you have two basic cases, isomers and isobars. Isomers are compounds with the identical exact mass. This is the case here for hesperetin and hematoxylin. They have the same raw formula, thus C16H14O6. And they got the same molecular monoisotopic mass, which is 302.0790 mass atomic unit. And then you've got the case of isobars. So they have identical nominal mass, but they have different exact mass. This is the case here for two pesticides, carbofuran and formetanate. They have the same nominal mass, which is 221, but their exact mass is different, and their raw formula is different. C12H15NO3 and C11H15N3O2. The precise measurement allowed by some mass spectrometers will allow to differentiate isobars, but it's not going to allow to differentiate isomers. For this, you will need to separate them with chromatography or to analyze their fragmentation patterns. Hopefully, they will make different daughter ions. Some of them might be diagnostic ions associated to one structure in particular. And also, there are other devices like ion mobility I won't explain here. So to determine the raw formula, you can use two tools. First, well, the exact mass, and also what we call the I-fit, or the use of the isotopic profile. So this is what is given by a mass spectrometer for a molecule here. You have an ion of 353. You might guess that it is the caffeoylquinic acid in negative mode because we've lost one proton. So instead of 354, it's 353. And so this is the measured mass. And then you have masses that have been calculated according to different raw formula. And one of these formula, the first one, is C16H17O9. But you also have other candidate formulas with very close exact masses, like, for example, C17, H13, N4, and O5. In this case, you have 4 atoms of nitrogen, and it gives you a very, very close mass to that that was measured. So, well, the first criterion is how far these two exact masses are. So, for example, the first formula is 1.7 ppm drifted from the measured mass, while the third raw formula, CH13N12O10, which is quite exotic because you only have 1 atom of carbon and 12 atoms of nitrogen, so it's very unlikely when you know organic molecules, but the difference in ppm is only 0.3. So for this criterion, this raw formula is closer to the measured formula, the measured mass, than the other one. So you need another tool to sort it out, and you can use the isotopic profile. So if you know the raw formula, you can calculate how should look the isotopic profile. So that is, what should be the abundance of the M+1, M+2, M+3 isotopic profile. And then you can calculate a match factor between the isotopic profile of the measured molecule and the theoretical isotopic profile of the different raw formulas. And then you have a fit confidence. And here you see that for the first formula, C16H17O9, you have 94% confidence, while you have 0% confidence for the third raw formula. So in this case, given that the two masses are very close, just 1.7 ppm away from each other, and the fit confidence is very high for this formula, you will probably make the reasonable hypothesis that this raw formula is the actual formula of the measured molecule. So exact precise mass and isotopic profile might give you a raw formula. It's very important but not sufficient, as we saw. And just to give you an example, if you have a request mass of 147 plus or minus 0.3 atomic mass unit, then the number of raw formula available is 13 in just one database, the human metabolome database. So you have many candidates, but now if you are more precise and exact on the measurement of this mass, so imagine you have, in fact, the mass is 147.262 plus or minus 0.003. Then you only have one raw formula in this database that can match your compound. And also, if you add up to this the isotopic profile, well, you can have a pretty good confidence in the decision to identify this molecule. However, it doesn't give you any structural information. So you may add another level of analysis, of structural analysis, with MS2 information. So we've seen the principle. You just break the molecule and analyze the fragments. Here you have two mass spectrum. One is the mass spectrum of the parent, and you see different ions, 343, which is M minus H, and also 191, which might be, if my memory is good, the quinic acid residue. So you have in-source fragmentation here. And you see the abundance of the base peak, which is 199, is 2 multiplied by 10 exponent 5. Okay, so it's quite intense. And then on the right side of the slide, you have the MS2 spectrum of the same molecule. And so you see that the total signal is much lower. You have maybe a reduction of 2 or 3 times. It's normal because you fragmented the main ions. But you see some fragments that I showed in blue that can indicate the structure of the molecule. So you can analyze this mass spectrum, MSMS or MS2 spectrum, to confirm the structure of the molecule that was given to you by a database based just on the exact mass and isotopic profile. So first, of course, you will use databases to find molecules with the same identical mass, and also with identical MS and MS2 spectra. So many databases are available. None of them is perfect. It's always a matter of reflection also. You need to think about your molecules and the options, and maybe sometime to use the literature to make you to have a firm opinion about the identity of your biomarkers. It's quite tricky. So we can mention MassBank, PRIME, ChemSpider, the Human Metabolome Database, MedFrag, and GNPS. GNPS is quite recent way, and we are still looking for the limitations of this technique, but it's very promising to identify new compounds. This is an ongoing process and a work in progress to use this MSMS information and the mass spectrum in general to identify clearly the molecules. It's critical to be able to to assess the risk you take by identifying a particular compound. So for this, there's been some work by a team, which was the Chemical Analysis Working Group and the Metabolomics Standard Initiative in 2007, and there's been another paper in 2014, which established the minimum reporting standard for chemical analysis. And regarding identification, they found 4 levels of identification: identified compounds, putatively annotated compounds, quantitatively characterize compound classes and unknown compounds. The first level is identify compounds. To say that you've identified a compound in a given analysis, you need two independent or orthogonal data that are relative to an authentic compound analyzed under identical experimental conditions. It means that you need the pure reference standard. You need the molecule analyzed almost at the same time than your sample. And from these analysis, you must have two information basically independent, like, for example, the retention time and the mass spectrum of the molecule, or the retention time and the NMR spectrum, or the accurate mass and tandem MS, which is MS data, etc., etc. So this is quite stringent. It's very hard to satisfy these conditions. So it's quite rare to identify formally a compound in the untargeted analysis. Then, much more common is the level 2 identification, which is a putative structure. This time, you just don't need the chemical reference standard. So you will use physicochemical properties and spectral similarity with public or commercial spectral libraries. So this is exactly the same definition as level 1, but you don't need to possess and inject in the same conditions the pure reference compound. You can use databases. So this is the level at which most metabolites in most studies are identified. So we can expect that there are many false identifications in those papers. Well, this is the state of the art, and this will improve with time and with the methods of identification. Then you can also just know the chemical family of a compound, because maybe its mass spectrum is very close to that of a known compound from the same family. Maybe you can say it's a sugar, because its mass spectrum looks like a sugar, because I find fragments that are very typical from sugars. Or you can say, well, it's probably a phenolic compound, because its absorption is in the range, well, is around 300 nanometers. And finally, you have unknown compounds. So from an experimental point of view, they can be important because they can be biomarkers, in fact. And it's also exciting to find biomarkers that nobody knows, because there is a massive amount of information. But it's very hard to identify de novo a compound, which means when it's a compound that nobody ever found in such sample. So in conclusion, and to summarize, you need the exact mass that will give you a raw formula, especially if you associate it with isotopic profile. And you will interrogate database to have candidate molecules. You can then practice MS2 fragmentation to find substructure or look into databases for to narrow the number of candidate molecules. You can use other spectral information like spectrophotometric information, absorbance in ultraviolet or infrared. You can have knowledge about the sample, especially chemotaxonomy is very important, because many chemical classes have been found in certain genera, especially when it concerns secondary metabolites. As I told you, they are specific to some alkaloids, are specific to some taxons of plants, like it's in papaver genera that you will find the morphinic alkaloids, for example. So if you know your sample, maybe you can have clues about the kind of metabolites you are supposed to find in this in these plants. Then there is a bibliography. If someone has found already this compound, maybe you can find it. And you can also make a full experiment to try to purify, concentrate, and analyze this compound with NMR and mass spectrometry, high resolution. This is long, expensive, and you have no guarantee that you will find, that you will in the end identify your compound. It's time now for the last chapter about to give some examples of metabolomics, especially in the fields of process and industry, even though there are many very interesting things to do in fundamental sciences. I will talk a bit about that with functional genomics at the end. So let's start with what we can call the foodomics, which is, well, the metabolomics applied to the food industry. So what can we do in this area? Let's start with this paper. It was published in Food Chemistry in 2014, and the title is Assessment of Fruit Juice Authenticity Using UPLC QTOF-MS, a Metabolomics Approach. So it's all about adulteration of juice. The European legislation states that an orange juice is made of pure juice of citrus sinensis, and pineapple juice is made of pure juice of Ananas cosmos. The Codex Alimentarius states that orange juice is the juice of Citrus sinensis, but up to 10% of tangerine juice, Citrus reticulata. And in the USA, the Food and Drug Administration states that orange juice is the juice of Citrus sinensis, plus up to 10% of tangerine juice, Citrus reticulata, and 5% of clementine juice, Citrus aurantium. So, well, the context is the fraud on the juices and the introduction of different quantities of other juices labeled orange juice. And this fraud has a cost in millions of dollars every year. So the experimental setup is the following. Juices were prepared from fruits, centrifuged and filtered, and there was a controlled addition of pineapple juice with 1%, 5%, 10 or 15% orange, apple, grape, or clementine juice. And then they made a non-targeted UPLC QTOF analysis of the juice and found markers defined by their, what we call features, which is a combination of retention time and mass as mass over mass to charge ratio. So this is a result in the form of a PCA, a principal component analysis. This is a score plot, which was obtained well in either positive or negative mode from orange admixture at 1% adulteration level. So you have different colors showing the different type of juice. So the pure pineapple is red triangles, and then you have 1% apple, 1% clementine, 1% orange, 1% pomelo, and another pineapple with 1% orange. Different kind of orange. And now this is an OPLSDA result on pure pineapple versus pineapple containing 5% of orange juice. So this is the S-plot and the loading plots showing important variables. You remember the S-plot on the top right or on the bottom left, you have the most discriminating variables, so metabolites. And finally, here is another graph showing the trend plots of 4 potential markers extracted from the S-plot. So these markers are shown in different colors, red, black, blue, and green. Each of them is a combination of retention time and mass to charge ratio. And, well, you see their abundance in different juices. So when you have pure pineapple, none of these markers is present. But when you have 15% of orange, then you have an accumulation at different degrees of these markers, where the red marker is less abundant in this case, but the green marker is a strong marker of orange in pineapple juice. Then you have pineapple with grape, and then you see that the most important marker is a black one. And finally, markers with clementine and orange. So this is the blue marker, which is the most interesting in this case. And then with pomelo, this is again the black marker. So the result here is just, you see, we don't even know what those metabolites are. All we know is that there are markers in the form of a retention time and a certain mass that are revealing the presence of different types of juices in pineapple juice. Then came the identification. So here you have the marker identity, the marker number, 1, 2, 3, etc. in the first column, its retention time, the ion, and then the identification of the type of adduct. So it's here in negative mode, and all of them are M minus H minus. And the fragments also. So they made MS to find fragments of these parent ions. And so they could calculate a probable putative ion elemental formula. They give here the mass error and the putative identification. So different types of compound that you may find in orange juice or clementine juice or grape juice, like narangine, hesperidine, etc., etc. Okay, so it was a very basic example on how to use metabolomics. So untargeted profiling, you extract features, and then these are biomarkers, and then you focus on these markers, and maybe you can imagine easily the application of this. Now we know what are the markers of grapes or orange or clementine. Maybe we can do some targeted analysis that will be easier to do on commercial juice to try to detect frauds. So here is a second example in the same kind. So it's targeted and untargeted metabolomics as an enhanced tool for the detection of pomegranate juice adulteration. It was published in Food in 2019. Pomegranate juice is called superfood. It's supposed to have a lot of nutritional interest and health benefits. It contains polyphenols, vitamins, antioxidant activity, etc., etc. There's been a strong growth of this market for this reason in the last 10 years. So it's tempting to adulterate these expensive juices with cheaper apple or grape juice. Even if you cut this pomegranate juice with 10% of apple, you make substantial economies. It's very important. So the idea is to find, once again, metabolic markers of adulteration. So they took 28 commercial fruit juices. 5 of them were 100% pomegranate juices. 8 of them were 100% apple juices, and 5 were 100% red grape juices. And also they made one fresh pomegranate juice themselves from a particular variety of pomegranate. They centrifuged and filtered, and they added controlled amounts of apple and grape juices, 1, 2, 3, 5, 10, 20% in the pomegranate juices. And they made both targeted and non-targeted LC-Q-TOF analyses. So here is the result for the targeted analysis. In the first column, you have the compound that have been searched and found. And then in the other columns, you have the amount that were detected in milligram per liter in the different type of juices. So the first is pomegranate juice, then pomegranate juice with another variety, apple juice, and red grape juice. And this is interesting just to find out what are the metabolites that are uniquely found in one particular juice. So for example, if you take caffeic acid, it's really a marker of apple and grape, to a lesser extent grape, because you have a high amount, 2.9, between 2.9 and 5.3 milligram per liter in apple, and only 0.045 or 0.012 milligram per liter in pomegranate juice. So if you find important quantities of caffeic acid, it means that there's been addition of apple juice. Then you have grape markers like catechin and hydroxytyrosol and resveratrol. So these grape markers are abundant, quite abundant, at least measurable in the red grape juice, but they are very weak or absent in the pomegranate juice. So when it is written inferior to LOQ, LOQ is limit of quantification. So maybe you can detect it, but you cannot quantify it. So the conclusion were that apple juice adulteration markers were epicatechin if it is superior, greater than 0.25 milligram per liter, which corresponds to approximately the addition of at least 3% of apple juice, or caffeic acid if you have a concentration in pomegranate juice greater than 0.36 milligram per liter. It means an addition of at least 5% of apple juice. Here is something we need to be careful, because in fresh pomegranate juice, they found epicatechin in much higher quantities than in commercial juices. So this is a limitation showing that depending on the preparation of the juice, depending maybe on the area of production or the process of sterilization, apatization, and so on, maybe you can find some epicatechin. And so this cannot be used as a marker. However, caffeic acid proved to be absent also in the fresh pomegranate juice and could probably be used as a marker for up to 1% adulteration. So on the bottom graphs, you have the chromatogram of epicatechin at different concentrations. So you see the size of the peak and the area of the peak is increasing the more you have epicatechin, and then you have a calibration curve. And finally, you have the mass spectrum showing the main ion of epicatechin, that would be the M-H-. Grape juice adulteration is also something you can monitor with epicatechin, at least 2% of grape juice if you have a concentration greater than 0.25 milligram per liter. Hydroxytyrosol or resveratrol if you find concentration greater respectively than 0.35 or 0.15 milligram per liter. And this means addition respectively of 3% of grape juice at least, or at least 20% of grape juice. So this data can be used to define thresholds of suspicion. So it means that if you make targeted analysis of these compounds in pomegranate juice, maybe randomly or maybe because you have an alert on some product, and you find content in these particular biomarkers greater than these thresholds, then you might want to investigate and look for possible traces of real adulteration and fraud. Now they also proceeded to a non-targeted analysis. So they analyzed the markers of apple juice, and here you have a table of the number of discriminant ions. So in apple juice and grape juice as compared to pomegranate, and at least 10 times greater in adulterant than in pomegranate. You have 214 apple juice markers and 191 grape juice markers. And so then you have the number of markers depending on the addition of the adulterant. So if you only have 1% of apple juice, you will have only 3 markers. And for 1% of grape juice also, you will have only 3 markers. But if you go up to 20% adulteration, you'll find 67 markers of apple juice and 47 markers of grape juice. So, in fact, this is more interesting than just the targeted analysis, because here you have a wider spectrum of potential markers to detect frauds. So they made some efforts to identify the most important markers. So on the right column, you have an indicative level of adulteration, 1%, 2%, etc., up to 20%. Then you have on the left column the identity of the marker, the number of the marker, 1, 2, 3, etc. The precursor ion, the retention time, the particular adduct of this compound, M-H- essentially, and the fragments. And then the probable elemental composition and the tentative identification, the putative identification. So here are some examples of MSMS identification of chlorogenic acid, which is an interesting marker because you can detect up to 1% adulteration. So it's a very low threshold of fraud detection. And so you have the formula of chlorogenic acid, and you see different mass spectra. So you have in-source fragments on the upper mass spectrum in red, and then you have MSMS fragments. You find some interesting ions like the dimer with a mass of 707. So it's 2M minus H minus. And then you have M minus H minus, which is 353. And you have a fragment of 191. And 191 is C7H11O6 minus. And this is the quinic acid part of chlorogenic acid, which is also named caffeoyl quinic acid. And so if you make an MSMS analysis, it is a blue spectrum, you will find other fragments, smaller fragments, that may come from sub-fragments of this molecule. They tried to classify the sample with a PLSDA, so it's basically the same than OPLSDA, but it's more, it's easier with many conditions, while OPLSDA sometimes is limited to 2 condition comparison. So they trained the model with 8 pure pomegranate juices and 30 juices adulterated with grape or apple. And the model test was 14 pure or adulterated juices. You remember that you train your model and then you test it. And they could successfully classify up to 2% adulteration. So this automatic classification of the sample allows to distinguish pure juice and juices adulterated with only 2% of adulterant. So here are the values of the cross validation of the model. So you have the R square, the goodness of fit, which is very high, 0.97, and the goodness of prediction, which is Q square, which is also very high, 0.93. Okay, so now let's see if we can do other interesting things with metabolomics in the industrial world. And this is a paper from 2018, published in Journal of Food and Drug Analysis, which is named The Integration of Multi-Platform MS-Based Metabolomics and Multivariate Analysis for the Geographical Origin Discrimination of Oryza sativa. Oryza sativa is rice. So the context is that you have different sources of white rice in Asia. White rice is a very important food. It's 20% of the world's calories, and it's 70% in East Asia. This is the equivalent of wheat in other parts of the world. You have 2 subspecies, Oryza sativa indica and Oryza sativa japonica. Japonica is mainly consumed by China, Korea, and Japan. So how can you control the origin of the rice, which is also likely to reflect its quality, the cultivation, the storage, and the pesticides that might be used in the different areas. So here is a problem of traceability and sourcing. In China, you find 19 varieties in 3 provinces. And in Korea, you have 13 varieties in 8 regions. So here is the experimental setup. They took rice samples, freeze-dried them, and stored them at -70 degrees. They extracted both polar and non-polar metabolites, and they made a GCMS analysis to find the primary metabolites and the fatty acids, and an LC cutoff to analyze phospholipids. I didn't mention it, but there is a whole field of analysis that is named lipidomics, that is attempting to analyze lipids, and obviously intact lipids, like lipids containing phosphate or ethanolamine and so on. These are the type of markers they decided to look for. So here are the results. First, a PLSDA model that reveals a very good potential for discriminating white rice sample between Korea and China. So it's more efficient with the LC data. You see that the separation is more clear. You have the percentage of variation shown by the different components. The first component expresses on the left 39.8% of the total data variance, and on the right graph, it's 28.3%. So the origin of the rice represent roughly one-third, at least one-third of the global variance, the global differences at the metabolic level here between the rice. Now you have box plots showing different concentration among the most discriminating markers. So the upper graph is relative concentration of sugars and sugar alcohol and fatty acid from the GCMS analysis. Well, it's not really obvious that you really have strong markers, maybe linoleic acid. And the other graph shows the relative concentration of phospholipids, and there you may have some interesting markers like lyso-PE 18.2. So this is the number of carbon and the number the place of insaturation, the number of insaturation. Now here is a PCA, so another multivariate statistics, a score plot and a heat map of sugar and sugar alcohols, fatty acid and phospholipids. And once again, it becomes obvious that phospholipids are the best metabolites to look for if you want to discriminate Chinese and Korean rice. So these phospholipids might be lysophospholipids, in fact, which are phospholipids having lost a fatty acid residue. So you have the intact phospholipid with two chains of fatty acid, and the lysophospholipid, which has lost one of these ramifications. So here are the different markers. Of course, there should be more statistics to validate these markers. It's not sufficient what they did. And also, it is quite strange, but they didn't report any information about discriminating the regions. So maybe they didn't find strong markers of the region. Maybe, in fact, their analysis or the whole paper maybe is a bit weak and flawed because it's weird that they didn't report markers for they were looking for. So it's a proof of concept that you may find useful to use metabolomics for sourcing, but maybe in some cases it's not that obvious apparently. Now comes another example to discriminate different foods depending on the process, the industrial process. So it's been published in Food Chemistry in 2019, and it's called Variation of Ceratonia silica pod metabolome in the context of its different geographical origin, ripening stages, and roasting process. So first, Ceratonia silica, which is caroubier in French, so it's a Fabaceae in the Mediterranean basin, and the fruits are seed pods. And it's widely used as an additive in the food industry. You have carob flour, which is E410. You find it in ice creams, pastries, syrups, chocolate substitute, and so on. And you have locust bean gum, which is Endosperm galactomannan, also called E410, and it's a thickener. So it's used in sauces and other industrial sauce and mayonnaise and so on. So the fruits were collected at 3 stages: green, turning, and mature, which is black, in 3 regions of Egypt: Bahira, Giza, and Siwa, and 3 replicates of 5 pods were taken. So the Siwa fruit were pulverized and heated to 120 degrees for 30 minutes to simulate a roasting process, which can be done in industry. Then they extracted the metabolites with methanol, and they made a profiling in GCMS for primary metabolites and LCMS for secondary metabolites. Let's say that they found 70 metabolites in GCMS. So they used TMS derivatization for primary metabolites, of course. And here is the effect of ripening. So you have unripe, ripe, and roasted Ceratonia silica pod collected in Siwa. So you can see some differences concerning maturity and roasting directly on the chromatogram, especially in the region of disaccharides, like sucrose, for example, or you have many disaccharides like maltose and so on. So here is the case for sucrose and maltose with the PCA score plot and the loading plot. On PC1 and PC2, you see PC1 is expressing 49% of the total variance and PC2 21, so you have a pretty much good view of the variance of the dataset on this principal component analysis. And in this case, you have the different origin, Giza, Siwa, and Beira, and also combined with different stages of ripening and roasting process. And so you see that sucrose will be a maturity marker. It separates the maturity. And maltose on the left part of the loading plots is rather a marker of immaturity. Then you have an OPLSDA to distinguish between roasted and ripe fruits. So the roasting results in a decrease in primary metabolites, especially sucrose. So it's also a marker of roasting. So it could be due to Maillard's reaction that involves sugars and amino acids during the roasting processes. Now, they also made a hierarchical clustering and principal component analysis of the secondary metabolites. They analyzed with LCMS. And in this case, they didn't discriminate between the geographical origin. This is what they show here. But it discriminated mature and immature fruits. So the marker in this case could be tetra-O-galloyl hexose, for example, that is a marker of immature fruits. Here is another OPLSDA showing the difference between unripe and ripe and ripe and roasted. So it shows, in fact, that if you, well, the secondary metabolites cannot really discriminate between the region, but, well, you can find, as I said, some markers of ripening and roasting. And in this case, you have the same, of course, than in the principal component analysis, but it's even more obvious. And you find tetra-galloyl hexose. So you have an enrichment of the less mature fruits in tannins and flavonoids, which is logical because this is what you would expect from immature fruits. They are protected from being eaten by these bitter and toxic compounds. And once the fruit is ready to be eaten for the seed dissemination, the tannins disappear and the taste is much better with sugars, and the values, nutrition value increases so that animals can eat the fruits. So what can we conclude from this? Well, here first, the limitations related to the genotypes is very important. You might find many markers between apple and pomegranate, for example, but using certain varieties. And if you change varieties, maybe some marker won't be valid anymore. So, well, this is a very important limit. And when trying to find a reliable marker of adulteration or traceability, it's very important to consider all the possible variation sources that might determine this discrimination, and genotype is one of them. And also we could add the type of cultivation techniques. Intensive cultivation or extensive cultivation will probably generate different metabolic profiles regarding the abundance of the compounds within the products. You also have limitations related to the process. So it's hard to get some standardized raw materials because depending on the industry, depending on the usage of the product, you'll have different storage methods, any kind of process from baking to freezing to crushing and so on, or even fermentations. And these are sources of variation that are not always easy to control in the field. So your sampling must be extremely careful. You have also limitations, as we saw, that are related to analytical choices. Which family of compound do you want to analyze? We saw that phospholipids were better discriminating different types of rices than primary metabolites. So it really depends on the machine you have and on the kind of analysis you do. So your development for chromatography and the choices you make will impact strongly the result. So in the end, maybe a positive remark is that the best markers are chemotaxonomic markers, which are extremely characteristic, as I already explained, of a variety, but most of the time of a species or a taxon, a particular genera or family of plants. So if you find an alkaloid it's not a very good example, but if you find certain types of phenylpropanoids in orange juice, and you know that there is not the pathway that can produce this phenylpropanoid in the orange, it's not in its genome. So then you can infer with pretty very high level of certainty that this orange juice has been altered and adulterated. So to apply really that to the world of fraud, you have a problem of reference. Which reference use would you use to train your models, your OPLS models or PLS models, and how to detect the effects of dilution? Sometimes you have thresholds in the machine. When you analyze a compound, you might not see it because it's too diluted. So in industry, it can be developed from targeted analysis rather than untargeted, but untargeted analysis can give new ideas about marker to look for in a targeted way. So we'll see now a short example of another application of metabolomics in a totally different way, which is the field of functional genomics. So it's a bit more fundamental research here. First, let's define what we call the post-genomic era. Well, we entered in the post-genomic era in 2000 when we first published the genomes of many model species. So you have bacteria, yeast, insects, mouse, and this is still going on, and we sequence more and more organisms. In the plant kingdom, the big breakthrough was the sequencing of the genome of Arabidopsis thaliana, which is the model plant. It's very practical to make research on plants. So it was published in Nature in 2000, and well, they found more than 26,000 genes, a few pseudogenes and non-coding RNA. But of course, it was already known, but the proof was there. At least 30% of the gene have no non-function. And so what we call functional genomics are all the research strategies aiming at link the genes, the sequence, to its biological function. So what protein is produced and what is the function, the biological function of this protein. Fortunately, at the same time, some progress were made in molecular biology, and we went from a situation where we could study genes in a forward manner by mutating randomly a high number of seeds, and then identifying the mutant showing an interesting phenotype, like a mutant with who was impaired in growth or enhancing growth or was more or less tolerant to one type of stress. So you had to make a screening of this mutant for a particular phenotype. And only after that you could map the genetic mutation and identify the gene that was responsible, hopefully, for this phenotype. And then with new tools of molecular biology and especially transgenese techniques, we went into a reverse genetics tactic. So in this case, you start with the genotype. You modify a gene because you have the sequence and you can do that. And you generate many lines that are mutants. They are knockout, overexpressor, silence, and you have collections of genetically contrasted lines. And then only, after selecting one gene, you are looking for its phenotypes. And this will make the functional annotation of the gene. So it's completely the opposite way around as before, and it's much faster and much easier to analyze the function of the gene this way. Just remember that phenotype, we call phenotype all the traits of an individual, its characters, like its morphology, its physiology, and also every kind of mutation it could have at the molecular level. And the phenotypes comes from the expression of the genome during the development of the organism and under the influence of environmental factors. It's a common mistake to believe that a phenotype only depends on the genome. But no, it's a genome in a particular environment. To be convinced of that, you have here a small experience. So you have two genotypes, a wild type of Arabidopsis and one that has homozygous mutation on one gene, that is LUT51. So it's a knockout. It cannot express this protein. And you see that under normal growth condition with normal light intensity, you see basically no difference between the two genotypes. But if you change the environment with photonic stress in high light, then you will see that the wild type is able to synthesize a pigment to protect him. And this is a carotenoid in this case, while the mutant is unable to do that, and it's bleaching instead of getting red or brown. So the phenotype is expressed, it depends, of course, on the genotype, but might be expressed only in certain environments. So, of course, the game is to produce many, many genotypes. So you have collections of ecotypes, cultivar mutants, genetically modified organisms. It's true both in plant and animal kingdom research. Then you made some experiments, and then you look for the phenotypes, the macroscopic phenotype, shape, size, productivity, yield in agronomy, but also more and more the molecular phenotypes, like the changes in transcriptome and proteome and metabolome. And this will give you more mechanistic information about what has changed. So here you have again the layers, the different layers of organization. And all this is connected into network. And so you will access different levels of this organization. So let's see a paper that is working in the functional genomic field. It was published in 2014 in Plant Physiology, and it's named Metabolomic Characterization of Knockout Mutants in Arabidopsis, Development of a Metabolite Profiling Database for Knockout Mutants in Arabidopsis. So the idea is they took a single background, a genetic background, which is named COL0. It's the most commonly used genetic background of Arabidopsis. And they made 50 Arabidopsis mutants from this background. And for each of them, they took phenotypic data related to the growth, the color, well, mostly by taking pictures of these mutants. And then they analyzed by GCMS for primary metabolites. They integrated 200 peaks and identified from these peaks 95 metabolites to make a database gathering together the phenotypic information and the metabolic information, and of course the genomic information, what gene was mutated. And they made this web portal called MECO, this database. So here you have the first set of graphs showing the macroscopic phenotypes, like the leaf color. You have a pale green for less than 10% of the mutant. You have a change in the leaf shape for less than 10% of the mutant. And you have a change in the root shape for less than 10% of the mutant. So it means that most mutants have no obvious phenotype here. And maybe the metabolic phenotype will be more discriminant. And so here is the global view of the interface of MECO. So you have the name of the gene that has been mutated, then you have the pictures and the description regarding the phenotype, and finally you have a metabolic map plotting the different metabolites that change between the mutant and the wild type. So here is the list of the genes and their functional description. For instance, ABA1-5, which is a mutant that is deficient in abscisic acid. which is involved in stomata closure, but also in hydric stress. And you have the name of the gene that is mutated, and you can click on it, and you will go on another website giving you a lot of information about this gene, especially you can get its sequence. Then you have the visual phenotype of the KO mutant. You see ET03 has a smaller growth than the wild type. And finally, you have here the metabolic map with a color code. The red metabolites are more abundant in the knockout than in the wild type, and the blue ones are more abundant in the knockout, are less abundant in the knockout than in the wild type. So you see that this mutant accumulates greater amounts of organic acids from the Krebs cycle or simple sugars like glucose and fructose, but accumulates less compounds such as phenylalanine and tyrosine, which are aromatic amino acids that paves the way to the biosynthesis of secondary metabolites. So this work is clearly just a proof of concept. Many metabolites are lacking, like you have no secondary metabolites, no hormones, and you only have 50 genes when we know that 26,000 genes exist. But this kind of work could, in the near future, in a few decades maybe, give rise to databases that we could look for to understand the function of the genes with unknown functions. So, because you would have the macroscopic phenotype and the metabolic phenotype, and you can make very educated hypotheses about the function of the genes that are investigated. And to conclude, we'll talk briefly about clinical diagnosis, but you will have another course by Jean-Charles Martin on this topic. I will just mention this paper from 2013, which is Metabolomic Profiling as a Useful Tool for Diagnosis and Treatment of Chronic Disease, focus on obesity, diabetes, and cardiovascular disease. So the idea is simple. You can take a biological sample from many people, urine, feces, mucus, cerebrospinal fluid, and so on, and make a whole range of analysis on these samples by gas or liquid chromatography, NMR, mass spectrometry of any kind, and of course use multivariate statistics to extract biomarkers. And by comparing the health status of these people, of these patients, and their metabolic profile, you can either find diagnostic markers, which can be used directly to diagnose people with a pre-diabetic state, for example, but you can also have an insight into the mechanisms that are working in the particular health issues. What makes a person diabetic, and what kind of metabolic changes are associated to diabetes. And of course, you can gather that in massive databases for health societies. So here you have an example of a PLSDA of people that are overweight, obese, or normal. So you clearly discriminate them easily on the first principal component, and you can find some biomarkers associated to obesity obtained from the plasma. And for example, you will find some amino acid or some lyso-phosphatidylcholine, phospholipids, aromatic amino acid, or carnitine esters. So it's, as I said, a good information to both fundamental research and personalized medicine. Maybe different obese persons have different metabolic profiles. Maybe the mechanisms are not exactly the same in all types of obesity. And so by diagnosing these markers in different obese people, maybe you can change the treatment and give a more adapted treatment. So this could be very powerful if you associate it to the detection of mutations. So this is what we call GWAS, Genomic Data Association. And so by combining GWAS and non-targeting metabolomics in this particular case, you might find statistical links between genetic mutation and metabolic phenotypes and pathologies. And so that's very powerful, because then you can make, you can think about it and you can investigate at all these levels of organization, from the gene to the pathology, which is the phenotype, in fact. And the intermediate being metabolic phenotypes, indicating some imbalance, for example. And you also have many other applications of metabolomics in medicine, especially in oncology, to detect tumors or to classify tumors, to know if they will be, to predict if they are going to respond well to a type of another of treatment, for example. So it can also make some personalized medicine, and this is a field that has been developing for 10 years now, and is still a growing field of interest regarding metabolomics. So this is the end of the course. Thank you for your patience.